يعطيكم العافية أحبائي وكل عام وأنتم بخير مرة ثانية. Today I will talk about the cerebrospinal fluid, which is the second body fluid after finishing urine analysis, routine urine analysis and the special test done for urine. Today I will talk about the cerebrospinal fluid. And cerebrospinal fluid, or CSF, as you know, this sample is represent the central nervous system. So the purpose of CSF analysis is to diagnose any disorder that affect the central nervous system. We have many conditions need to take this sample. Some of these conditions, for example, parasitic infection, fungal infection, or viral infection, or bacterial infection that cause meningitis or encephalitis. Also, if the patient having a tumor, cancer of the nervous system, we need to take the CSF sample. In some cases, also, if the patient have a sexual transmitted disease, syphilis, we need to take this sample. Bleeding or hemorrhaging around the brain of cerebrospinal spinal cord, we need to take this sample. Also, in case of multiple sclerosis, disease that affect the myelin coating of the nerve fibers of the brain and the spinal cord, in this case also we need to take the CSF sample. Now about the collection and transportation. Collection always done, as you remember, by physician under sterile operative condition. وهاي العينة لازم تتاخذ بسبيشال physician, because this sample is very sensitive. You cannot take it any time during the day, like blood agar media, or like urine sample, or like stool sample. For that, this sample is considered very expensive and very and emergency sample. When you have many samples in the lab, always analyze the CSF sample, because this sample is considered as emergency sample. يعني لما يكون عندك مجموعة من العينات في اللاب، دائما بنعطي الأولوية لعينة اللي هي CSF. الcollection, as you remember in diagnostic microbiology 2, I will remind you about the collection. The patient must lie on his side with the flexed knees and arched back to separate the lumbar vertebra, as you remember, and head and chin bent down, knees drawn up to the chest, and arms clasped around the knees. زي ما شفنا الحركة انه كيف حاط الايدين حوالين الركبة هيك مشان يعمل اللي هو separate لللمبر vertebra. هلا السبيس between lumbar vertebra L3 and L4 for adult, or L4 and L5 for children, is palpated by 4 finger. زي ما حكينا بنصير ندور على اللي هي المنطقة ما بين الفقرات. بس نلاقي اللي هي السبيس between lumbar vertebra 3 and 4 for adult أو lumbar vertebra 4 and 5 for children. شو بنعمل اللي هو لازم نعمل اللي هو disinfected للسكن area by using اللي هو 70% of isopropyl alcohol following by 2% of provodone iodine. هلا الفيزيشن must wear sterile gloves. زي ما حكينا انه الفيزيشن لازم يكون لابس sterile gloves، وممكن نستخدم هنا اللي هو الان injection of local anesthetic حتى to make the collection easy على مكان اللي هو البنكتشر سايت. هلا السبايدر نيدل عادة احنا شفناها قديش طويلة is carefully inserting directly into the spinal canal، وحكينا until pop is filled or until CSF is flow. بضلنا ادخل في النيدل حتى CSF الفلويد يبلش يطلع من النيدل. هلا مجرد ما وصلت دخلت النيدل وصار CSF flow، لازم اللي هو اقيس اللي هو البريشر في السباينال كانال من خلال اللي هو اللونج ثين تيوب اللي سميناه المونوميتر. بعمل له اتاج على النيدل وبقيس اللي هو البريشر لهذا الفلويد. هلا الفلويد should be collected زي ما حكينا and dispensed on 3 وبعض الأحيان 4 screw capped tubes. هلا الليبل بعد ما أجمع العينات، العينات دائما تتعبى sequentially. التيوب number 1 I will label it with number 1. Tube number 2 I will label it with tube number 2. So the label is very important for the tube. The name of patient is very important. A date, a time of collection, and the number of a tube should be reported on the tube. It's very important to fill it sequentially. بعد ما قديش عادة احنا بناخذ في كل تيوب. each tube should contain 2 up to 3 ml إذا كان عندي adult and 1 ml for children. Finally, the specimen transport immediately to certain lab, as you remember, to microbiology lab, to biochemistry lab, to cytology lab. Sometimes the tube number 4 we need it for serological or if the causative agent if the causative agent is Mycobacterium tuberculosis. And as you remember, in microbiology lab we need this tube to perform gram stain, acid fast stain, India ink stain حتى إذا بتتذكروا الكريبتوكوكس نيوفورمانس. Also, the first step in microbiology is to culture the sample in the primary place, as you remember, blood agar media, chocolate agar media, and makonki agar media. After culturing, you can perform the gram stain, you can perform acid fast stain or India ink stain. Because as you remember, CSF is sterile. There is no normal flora present in the sample. In biochemistry lab, we have two parameters need to screening for it, glucose and protein. In cytology lab, always we need to count the cell, the white PC and RBC, and we need to make the differential stain. If the white PC, the number of white PC is high, we need to know the type of leukocyte that is present in the sample. Now in this slide I will talk about the examination of the CSF sample or cerebrospinal fluid. The first part of examination, which is the physical examination or macroscopic examination, which is done with naked eye. Here we need to look for the color. Normally, color of CSF or cerebrospinal fluid is colorless. There is no color for the CSF. It looks like water. But in pathological condition, you can see it yellowish. Yellowish xanthochromia, elevation of protein, elevation of bilirubin. You can see the CSF اللي هو yellow. إذا بتتذكروا الأطفال حديثي الولادة دائما مشاكلهم بكون عندهم اللي هو الإصفرار، البيليروبين عالي. If you take the CSF sample, if you newborn, ممكن ألاقي اللي هو CSF لونها yellowish. Also in pathological condition you can see the CSF bloody. Bloody maybe due to two reasons: injury, due to the injury of by a needle that used to collect the sample, or due to brain hemorrhage or hemorrhaging in the brain. وحكينا بقدر أميز بين الانجري وبين البرين hemorrhage or bleeding in the brain. Bleeding on the brain you can in the brain, sorry, you can see the blood in all the tubes, but injury you can see the blood on the first tube. Maybe you can see it also in the second tube, and third tube, and tube number 4 you cannot see it. Now about the viscosity, there is no viscosity for CSF. Always watery, like water. ما فيها viscosity. The specific gravity you can use the refractometer, and the specific gravity for CSF 1.001 up to 1.008. high normal. What about the transparency? You can check as you check the urine sample. We need to using, for example, a printed paper or a white paper with a black line, and you need to place the sample in the front of this paper, and you can read through the sample what behind it. For example, the appearance of CSF sample clear. That means you can see through the sample. You can read what behind the sample very clear. هاي بنسميها clear sample. الhazy sample you can read from the sample, but not so clear as in the clear sample. The cloudy sample you can see through the sample, but it's very difficult to read. يعني أنا بقدر أشوف من خلال السامبل what behind the sample. You can see the paper through the sample, but you cannot read very easy. But you can read it what written in the paper. Turbid sample you cannot see through the sample completely. You cannot see through the sample what behind it completely. يعني ما بتقدر تشوف من خلال السامبل شو الإشي المكتوب على الورقة مسميها اللي هي turbid. وزي ما حكينا النورمال CSF always clear. For clot formation or pellicle يقول لك clot formation أو البيليكل اللي هي indication for the presence of fibrogen in case of mycobacterium tuberculosis. إذا كانت المننجيتس أو المننجيتس caused by mycobacterium tuberculosis, you can make اللي هو الأسيد فاست sustain from the بيليكل. إذا بتتذكروا كنا ناخذ tube number 4, place it at room temperature for 30 minutes, or you can place it in the refrigerator for 12 up to 24 hours. If there is any formation or membrane developed in the surface of this tube, that this layer indicate the presence of mycobacterium tuberculosis, and from this layer you can make the culturing, you can make the acid fast stain. الميكروبيولوجيكال examination عادة أهم إشي بالميكروبيولوجي examination to culture the sample on the primary plates. And the primary place that used to culture the CSF sample is a blood agar media, chocolate agar media, and makunki agar media. And remember that when you culture the chocolate agar media, this plate should be incubated under CO2 at temperature 35 or 37 under CO2 condition. Now, what is the suspected organism that cause the الله أكبر الله أكبر استنى بس يخلص الأذان نستكمل اللي كنا نحكي فيه بعد ما خلص أذان. إذا most common microorganisms that involved in meningitis اللي هم الشيريشيا كولاي, ستريبتوكوكس أجالاكتيا, نيسيريا meningitis, ليستيريا مونوسايتوجينيز في حالة الاميون سبريس بيشنت, الستريبتوكوكس نيمونيا, والهيموفيلس انفلونزا في حالة اللي هم الأطفال. هاي اللي هي الموست كومن مايكرو اورجانيزم. هلا في الميكروبيولوجي لاب شو حكينا؟ اهم شيء ان نبدا في عمليه الكالتشرينج اوف ذا سامبل. Sometimes if the causative agent is mycobacterium tuberculosis, in this case we need to add Lowenstein-Jensen agar media to the primary place to culture it. After culturing we need to perform acid fast sustain. For example, if tube number 4 have pellicles or have layer on the surface, after place it in the room temperature or after placing it in the refrigerator, from this layer you can make the acid fast sustain for detection the mycobacterium tuberculosis. Also capsule stain, which is the negative stain, you can use here nigrosine, or you can use India ink, you can use eusine to perform this test. And negative stain you can prepare it by mixing 1 drop of the CSF sample with 1 drop of negative stain like eusine, for example, mixing very well at one end of a clean slide. Then you can spread it as you spread the blood film. Under microscope you can see the microorganism surrounding with a huge capsule, colorless capsule, and this capsule stain or negative stain used to detect Cryptococcus neoformans, because as you remember Cryptococcus neoformans has a huge capsule. Also we can do wet mount preparation. Wet mount preparation by taking the CSF sample in the center of a clean slide, then cover it with cover slip, and you can examine it under microscope under 40x to see the amoeba. Also a gram stain is very important to perform it, because any organism present in the CSF and see it under microscope by using a gram stain is significant. ليش significant؟ لأنه there is no normal flora present in the CSF. So any gram positive, any gram negative present in your gram stain prepared from the CSF sample is significant, as you should report it by telephone to the physician. الchemistry examination عادة في عندي 2 parameters أنا بطلع عليهم في اللي هو الchemistry اللي هم البروتين والجلوكوز. In all case البروتين will be increase, the glucose will be decrease. The glucose always decrease except in the viral infection you can see it normal. What about the cell counting? I will talk about it in the next slide. Here in this slide I will complete the examination. and I will talk about the cell counting. And cell counting here, we need to count the white BC. Normally we count the white BC. Sometimes RBC is present, also we need to count it. But the counting of cell here depends on the appearance of the CSF sample. For example, if the sample is clear, in this case, that's mean clear. That's mean the number of white BC or the number of cells is very low. For in this case, we need to count the 9 square that is present in one area. In حكينا one area of chamber contain of 9 squares. If the sample CSF is clear, we need to count all 9 squares. إذا في حالة إذا كانت العينة CSF clear لازم أعد التسع مربعات الموجودة في المنطقة. يعني احنا حكينا كل منطقة في الجامبر composed of 9 squares. In this case, we need to count all 9 squares. There is no need to make the dilution for this sample because the number of white BC here is very low. So the dilution here will be 1. There is no dilution. In CSF sample, we have a special chamber used to count the cells. We called it Fox horizontal counting chamber. And the difference between this chamber and the common chamber, counting chamber, on the depth. The common counting chamber depth 0.1 as you remember, but in this Fox horizontal counting chamber the depth is 0.2. الفورمولا اللي بستخدمها to count the total cell count اللي هو as you remember cell count ضرب dilution factor multiplied by the depth factor divided into the number of square counted multiplied by area. يعني السل اللي انت بتعدهم في التسع مربعات multiplied by dilution factor. Dilution factor, there is no dilution here, so you can write 1. The depth factor, it depends on the chamber. The depth factor, in case if you use the common chamber, the depth of this chamber is 0.2. So the depth factor will be the opposite اللي هو the depth 1 على 10. 1 على 10. لما نقول factor المقلوب. So depth factor for common chamber is 10. For the Fox horizontal chamber the depth factor is 5. ليش 5؟ لأنه 2 على 10 depth. لما أقول لك factor مقلوبها شو مقلوبها؟ 10 على 2. 10 over 2. 10 over 2 means 5. So depth factor here is 5. بنقسم على number of square counted and the number of square counted here is 9. Multiply by area. The area for each square is 1. فبالتالي your result will be per cubic millimeter or per microliter. هلا السيل زي ما حكينا ال appearance of the CSF sample بيعتمد على اللي هو قديش عندك سيلز، قديش عندك وايت بي سي، قديش عندك ار بي سي، هو اللي بخليها تيربد او كلير او هيزي او كلاودي. اذا الكلير sample no need for dilution. When you have a clear sample we need to mix the sample 5 to 10 times by inversion. You need to charge the chamber or the hemocytometer. Then you need to count one area of the chamber. You need to count the 9 square and your result will be per cubic millimeter. And remember the depth of the counting chamber. If you use Fox horizontal chamber, the depth factor is 5. If you use the routine counting chamber, the depth factor is 10. خلينا ننتقل للسلايد اللي بعديها. والله قبل ما ننتقل للسلايد اللي بعديها في عندي في المانيوال تبعكم اللي هو مكتوب count all 9 square for white BC only by using glacial acetic acid. There is no need to use the glacial acetic acid to eliminate the RBC. Why? Because there is no dilution used for this sample. So please remove this sentence. By using glacial acetic acid to eliminate the RBC, remove it because there is no need to dilute this sample. When you have a clear sample, we need just to mix the sample and to charge the hemocytometer and to count directly without using the glacial acetic acid. عشان هيك please شيلوا هاي الفقرة by using glacial acetic acid to eliminate the RBC لأن أصلا ما بنعملش dilution هون إحنا بالغلط يعني جاية. Also here in cell counting if the sample abnormal appearance, having, for example, turbid or hazy or cloudy, in this case we need to make the dilution. And the dilution here we need to take to use the glacial acetic acid. How can I prepare the 3% acetic acid؟ كمان هنا في شوية تعديل على المانيوال اللي هو ال 3% acetic acid. إني باخذ 3 ml of acetic acid mixing by 0.2 gram of crystal violet to stain the cells and adding 97 ml of distilled water. هيك بكون أنا حضرت 3%. اوكي 3 ml glacial acetic acid mix with 0.2 gram crystal violet mixing with 97 ml of distilled water. بكون أنا محضر هيك ل 3% glacial acetic acid. So here in abnormal appearance we need to mix the sample by inversion. Don't shake the sample, we need to mix it by 5 to 10 times by inversion. Then dilute the sample by using the 3% glacial acetic acid. Now the dilution depends here on the appearance of the sample. For moderate cell count means if the sample is hazy or cloudy we need to make the dilution 1 to 10. هذا بنسميها المودريت. إذا المودريت هي بتمثل represent الهيزي وبتمثل اللي هي الكلاودي. If your sample is turbid we need to make the dilution 1 to 20 sorry. We need to make the dilution 1 to 20. إذا الهيزي الكلاودي we called it moderate cell count. In this case we need to make the dilution 1 to 10. If the sample turbid, in this case we need to make the dilution 1 to 20. Now in both cases we need to count the 5 square in both sides of chamber. The 5 square, as you see here in the picture, the 5 square اللي هم the 4 white PC square that present in the corner, and on the middle the white PC square, the RPC square. So 4 white PC square which is located in the corner of large square that composed of 9 square. إذا ال 9 square أنا بدي أعد منهم 5. ال 4 squares for white PC that located in the corner, and the RPC square which is located in the center which composed of 25 small square of RBC. إذا بدي أعد ال5 مربعات وبدي أعدهم in both sides of the chamber. So totally we need to count 10 square. 8 square for white PC in both sides and 2 square for RBC in both sides. Okay, بس أعد بطبق على القانون. Total cell count equal cell counted in 10 squares multiplied by the dilution. If you use the dilution, for example, 1 to 10, so the dilution factor is 10. Multiplied by depth factor. The depth factor, if you use the common counting chamber, the depth factor here is 10. If you use, for example, Fox tensile chamber, the depth factor here is 5. Divided into, بقسم على كم? Number of square counted. The number of square counted 10 squares. And the area of each square is 1. So 10 multiplied by 1. And your result will be per cubic millimeter or microliter. هاي بالنسبة إذا كانت الابيرنس اللي هي abnormal, moderate أو turbid. بدي أعمل dilution ل 1/10 إذا كانت moderate أو dilution 1/20 إذا كانت turbid. ولازم you need to count the 10 square 5 square in each area. وزي ما حكينا 5 مربعات اللي هم 4 تبعات الوايت بي سي، واللي بالنص اللي هو الار بي سي الخامس، وبروح على الجهة الثانية من التشامبر وبعد كمان 5 مربعات، بصير totally count 10 square. Now here I will talk about the cell counting in case if you have a bloody sample. In a bloody sample we need to do the correction of leukocyte count in the CSF. Now in a bloody sample, in this case we need to count both RBC and white BC. Bloody sample means that the RBC is present, and normally there is no RBC in the CSF sample. So when you have bloody sample, in this case we need to count both RBC and white BC. RBC, we need to count this RBC in the middle square or on RBC square in the 5 square that present in the RBC square. The 5 square, as you remember, 4 located in the corner and 1 located in the center. So when you have a bloody sample, we need to count the white BC alone, okay, and we need to count also the RBC in the middle square in the 5 square of RBC squares. In this case, remember that when you have a bloody sample, the blood coming from the peripheral blood, or the peripheral blood enter your CSF sample. And CSF sample here, when the blood come to the CSF sample, the white BC will become to your CSF sample, mixing with the white BC that present due to infection. يعني انت لما دخل عندك البلد، دخل وايت بي سي اختلطت مع الوايت بي سي اللي موجودة as infection في عينة CSF. The same time RBC also is enter your CSF sample. اوكي، معناته لما تكون عندك بلادي CSF sample معناته RBC دخلت على العينة CSF وخلتها بلادي، بنفس اللحظة دخل معها وايت بي سي. فأصبح هون لازم نعمل correction، لازم نعرف you need to know the white BC that come to your CSF sample from the blood sample or from the peripheral blood. We need to know the number of these white BC that come from peripheral blood and to remove it from the CSF present in the CSF sample. So when you have a bloody sample, we need to count the white BC and RBC. Now the number of white BC you have due to counting the CSF sample, the number of white BC is mix white BC. هذول الوايت BC اللي present in CSF in a bloody sample, some of them due to infection and some of them due to peripheral blood. We need to remove the white BC that come from peripheral blood. We call this cells اللي هم الوايت بي سي added. So white BC added means the white BC that come from the peripheral blood. We need to remove it from the total count of cell of white BC in the CSF sample. Remember that when you collect the CSF sample, in the same time we need to collect the blood sample. So for that, when for any patient for meningitis, you have 2 samples. One of them a blood sample, and the second one CSF sample. The blood sample we need to do the CBC to know the number of white BC, to know the number of RBC. Now CSF sample, when you have a bloody CSF sample, we need to count the white BC in the white BC squares, and the number of white BC here represent the white BC due to infection plus the white BC that come from the peripheral blood, واللي سميناهم white BC added. Also we need to count the RBC. Now to know the white BC that come from the peripheral blood, we called it white BC added. We have 2 formula. The first formula, the white BC added equal بتساوي white BC in the peripheral blood, you can know this number from the CBC, multiply by the RBC you counted in the CSF. What you count in the CSF of RBC, I will multiply it by white BC that come or that counted in the peripheral blood in CBC, divided into the RBC in peripheral blood. So from the CBC you can know the number of white BC in peripheral blood, you can know the number of RBC in the peripheral blood. So when using this formula, you know the white BC added. For example, if you count, for example, 100 cells of white BC in the CSF, you have CSF sample, mixing, making the dilution, and the number of white BC in this CSF sample is 100, but the sample here is bloody. So these 100 cells of white BC not represent the infection. We need to know the white BC added. So when you apply this formula, white BC added equal to white BC in peripheral blood multiplied by the RBC in CSF divided into RBC in peripheral blood, you know the number of white BC added. White BC added, suppose that for example 30 cells. معناته الوايت بي سي added that come from the peripheral blood is 30. In this case, الوايت بي سي due to infection equal to white BC that count in white BC in CSF sample, which is 100 for example, minus white BC added اللي هم 30. So the real number of white BC due to infection is 70. معناته خلينا نعيدها بالعربي حتى توصل أكثر. إذا عندك عين CSF بلدي بعد الوايت بي سي، بعمل ديليوشن بعد الوايت بي سي، بستخدم الجليشال اسيتيك اسيد بعد الوايت بي سي بعد الار بي سي. نفرض ان الوايت بي سي طلع عندك 100 وايت بي سي، هذول في عينة بلادي سي اس اف مش كلهم بيمثلوا الانفكشن، بعض منهم موجود نتيجة انفكشن والبعض موجود نتيجة انه دخلوا مع البريفرال بلاد. اوكي، دخلوا مع البريفرال بلاد واللي سميناه الوايت بي سي ادد. شو بعمل انا؟ بكون عامل عندي عينة بلاد، كنت عامل السي بي سي، عرفت الرقم تبع الوايت بي سي الموجود في عينة الدم، وعرفت رقم الار بي سي الموجود في عينة الدم، واوريدي الار بي سي انا عادتهم في السي اس اف قديش. اوكي، فبطبق الفورمولا الوايت بي سي ادد شو بتساوي؟ الوايت بي سي بالبريفرال بلاد قديش طلع عندك الوايت بي سي لما عملت سي بي سي، قديش طلع عندك الوايت بي سي؟ بحطه بضربهم في الار بي سي اللي طلعوا معي لما عديت الار بي سي، بضربهم عفوا في الار بي سي لما عديت انا السي اس اف طلع عندي ار بي سي، بضربهم في الوايت بي سي اللي جايات من البريفرال بلاد. هذا الرقم بقسمه على كم؟ على الار بي سي اللي طلعتهم انا من اللي هي السي بي سي البريفرال بلاد، بيطلع عندك الوايت بي سي ادد. نفرض انه الوايت بي سي ادد طلع عندي 30. إذا كنت أنا عاد الوايت بي سي بالسي اس اف 100، شو بقول أنا؟ الوايت بي سي للإنفكشن تساوي الوايت بي سي بالسي اس اف، كنت عاد فور اكزامبل الوايت بي سي بالسي اس اف 100، بنقص منهم الوايت بي سي ادد اللي هم 30 اللي طلعتهم بالفورمولا اللي فوق، صفى عندي الوايت بي سي ديو تو انفكشن اللي هي 70. هذول اللي موجودات نتيجة الانفكشن. أو ممكن استخدم other formula واللي هي بتقول لك 1 ليكوسايت مقابلها 500 اريثروسايت. اوكي، كل وايت بي سي، كل حبة وايت بي سي، كل سيل من الوايت بي سي بقابلها 500 ار بي سي في عينة البريفرال بلاد. مش احنا من دم بنشوف الار بي سي بالملايين، بينما بنشوف الوايت بي سي بالالاف. عشان هيك بقول لك كل واحد وايت بي سي مقابله 500 ار بي سي. فبطبق الفورمولا هاي. نفرض على سبيل المثال ان عدينا الار بي سي في عينة السي اس اف مثلا 30,000، عديت الار بي سي في عينة السي اس اف والنمبر اللي هو 30,000. اوكي، معناته when you count the RBC in the CSF, suppose that the number is 30,000 cells per cubic millimeter. اوكي، هلا بدي اشوف هذول 30,000 قديش دخل معهم وايت بي سي. بطبق الفورمولا. اوكي، 1 وايت بي سي مقابلها 500، قديش مقابل ال 30,000؟ بضرب ضرب تبادلي بيطلع اللي هو الوايت بي سي ادد. سو الوايت بي سي ادد هير بعملها، بشيلها من الوايت بي سي اللي في السي اس اف، بيطلع عندي اللي هو الوايت بي سي بالانفكشن. اوكي، هلا بكتب لكم سلايد مشان اوضح لكم العمليه. Here I will talk about the bloody sample. If you have a bloody sample, we need to make the correction. For correction we have two formula as I mentioned in the previous slide. Example for the first formula, and you remember the first formula say white BC added equal to white BC in peripheral blood multiplied by RBC in CSF divided into RBC in peripheral blood. For example, suppose that you have patient and you have two samples for this patient, one of them a blood sample and the second one CSF sample. From blood sample we can do the CBC to know the number of white BC. Suppose that the number of white BC for this patient in the blood sample is 9,000 and RBC is 5 million cells. RBC. And in CSF sample, when you count the white BC, you have 100 cells of white BC. But as I mentioned before, 100 is present as a mix from peripheral blood and from the CSF that present as infection. يعني الوايت بي سي لنعاتهم بالسي اس اف 100 بس مش كلهم ال100 نتيجة الانفكشن، في منهم نتيجة انهم دخلوا من البريفرال بلد. الار بي سي في السي اس اف عديت انا طلعت 30,000. اوكي، معناته هاي المعطيات اللي عندي. So we can apply this number on the first formula. White BC added that come from the peripheral blood. You need to know this number. So the number of white BC that come from peripheral blood multiplied by RBC in CSF divided into RBC in the peripheral blood. White BC added تساوي 9,000 multiplied by 30,000 divided into 5 million. So you have the number of white BC added to the CSF sample. So the number of white BC that come from peripheral blood is 54 cells that come from peripheral blood enter the CSF and mix with the white BC that present to you to infection. Now, when you know the number of white BC added, so the number of white BC that present in the CSF due to infection, we can know this number by white BC that count in CSF minus white BC added. So the number of white BC in CSF is 100. So you can remove 54 cells of white BC added that come from peripheral blood. So the total number of white BC that due to infection is 46 cells. هذول ال 46 cells هم نتيجة الانفكشن. إذا لما تكون عندك عينة بلادي سامبل وتعدي الوايت بي سي تلاقيهم 100 وتلاقي في ار بي سي وتلاقيهم 30,000 مثلا، معناته 30,000 هذا دخل معاهم وايت بي سي واختلطوا مع الوايت بي سي تبعات الانفكشن. كيف أعرفهم؟ بطبق الفورمولا الوايت بي سي ادد. قديش بيطلع وايت بي سي ادد عندي؟ بنقصهم من التوتال وايت بي سي اللي عديتهم أنا بالسي اس اف. فطلع عندي مثلا 54 سيل ادد، نقصتهم من 100 اللي كان توتال بضموا البوث، فطلع عندي الرقم due to infection اللي هو 46 cells of white BC. هذا example for the first formula. السلايد الثاني راح اعمل example for the second formula. Now second formula, example for second formula in case of you have a bloody sample. Each white BC مقابلها 500 of RBC. يعني كل white BC بقابلها 500 RBC. Suppose that you have CSF sample. At that the number of white BC that counting in the CSF sample is 500 cells, and the number of RBC that counting in the CSF sample is 50,000 cells. In this case, I will apply this formula. White BC added. Each white BC مقابلها 500. قديش مقابل 50,000؟ Okay, the number of white BC in the second area we have 500 of RBC. Now if you have 50,000 of RBC, how many cells of white BC come and enter the CSF from the peripheral blood? We need to know this number, and this number we called it white BC added. نضرب ضرب تبادلي. So white BC added equal to 50,000 of RBC divided into 500 cells. So the number of white BC added is 100. So white BC that present due to infection equal to white BC and CSF minus white BC added. قديش الوايت بي سي اللي عديناهم بالسي اس اف؟ 500. بنقص منهم الادد اللي طلعته مع الفورمولا هاي 100، بيطلع السول وايت بي سي due to infection is 400 cells، بيطلعوا اللي هو 400 cells. اذا ال 500 مش كلهم كانوا نتيجة الانفكشن. Not all of them due to infection. We have only 400 cells due to infection, and we have 100 cells that come from peripheral blood enter with the RBC. So the real number of white BC due to infection is 500 in this case. Okay. Here I will talk about differential count. And differential count is very important to detect the type of infection. If the meningitis due to bacterial infection or viral infection, it's very important to know the type of infection. وهذا كثير بيساعد في عملية التريتمنت لهذا البيشنت. So to do the differential count, we need to mix the sample 5 to 10 times by inversion. Then you can centrifuge the sample at 2000 RPM for 5 minutes. After that, what we need to take the sediment, not the supernatant. We need to remove the supernatant and to take one drop of sediment and place it in the center of a clean slide. And this drop that present in the center of a clean slide, we need to spread it 1 centimeter by 1 centimeter as a square. As you spread the blood film, we need to spread it in a small area to make the screening of white BC very easy. After you spreading the sample 1 centimeter by 1 centimeter, you can spread it by cover slip, for example, and keep the smear until dry, and you can fix it by using methanol. You can add or cover the slide with methanol and keep it for 5 minutes or 3 up to 5 minutes. After that, you can stain the slide with Giemsa stain or Wright stain. The Giemsa stain, you can add it for the slide or over the slide and keep it for 10 minutes. Then you can wash the slide with tap water to remove the excess amount of Giemsa stain and dry the slide. Then you can examine it under oil immersion lens. What you see under oil immersion lens, you can see the white BC. If the number of white BC present is, or if the type of white BC present is neutrophils, that means the infection is due to bacterial infection. If the cells present is lymphocyte, all of them lymphocyte, that means the infection is due to viral infection. Then the differential count is very important to detect the type of infection. This is the latest slide show you the normal findings of CSF. The CSF color, as I mentioned before, normally the CSF is clear. Glucose, the concentration of glucose normally present in the CSF between 45 up to 80 milligram per deciliter. When the glucose is decreased, that means is infection. When the glucose is normal, that means some case for viral infection, then the glucose you can see it as you see as 45 up to 80. يعني no change in glucose when you have viral infection. While in protein, the protein will be increased. Normally the protein present in the CSF between 20 up to 45 milligram per deciliter. The CSF pressure بيكون from 50 up to 80 milligram per milligram Hg. The normal CSF, the present of white VC leukocyte count in normal CSF 0 up to 5 cells per cubic microliter in case of adult, in case of children 0 up to 10, and in case of a newborn or units 0 up to 30 cells of per microliter. There is no neutrophils present as normal in the CSF sample. يعني ممنوع يكون عندك neutrophils في عينة السي اس اف. موجود عند الليكوسايت بنسب معينة زي ما حكينا في حالة الادلت، مسموح يكون من 0 up to 5، بس مش مسموح يكون في منهم neutrophils. There is no neutrophils present normally in the CSF sample. The normal count of leukocyte for adults 0 up to 5, for children 0 up to 10, for units or اللي هو حديثي الولادة بتكون 0 up to 30. And most of them are lymphocyte. 90% of these cells are lymphocyte. Okay. إذا مش مسموح يكون عندي neutrophils في عينة CSF. If you have one cells, if you have for example, if the number of white BC in adult 5 cells and you have in differential count 1 cells in neutrophils, that means the patient is infected with bacteria because there is no neutrophils present in the CSF. Any cells of neutrophils present in the CSF sample indicate infection or indicate bacterial infection. هلا 0 to 5 cells that present in adult, most of them are lymphocyte. 90% of them lymphocyte. 0 up to 30 cells of white BC that present in unit, 90% of منهم اللي هم lymphocyte. There is no neutrophils present in this type of specimen. هيك بكون خلصت CSF وإن شاء الله نكون استفدنا منها يا رب.